Coming to the next important reaction, high yield reaction is PFK1, phosphofructokinase number 1. What you should know about this enzyme? First statement, it is the committed and most regulated step in glycolysis. What it means? Committed step in the sense, let me take you back to this slide here. See this is where your enzyme, that is enzyme number 2, phosphofructokinase number 1, PFK1. It converts fructose 6-phosphate into fructose 1,6-bisphosphate. Why this is called as a committed step for glycolysis? That means once you convert fructose 6-phosphate into fructose 1,6-bisphosphate, the only direction that your fructose 1,6-bisphosphate will go from now is glycolysis only, to pyruvate only. It's not going to go another direction. It cannot go to pentose phosphate pathway, glycogen synthesis, glycoprotein formation. It cannot go that way. And that is why PFK1 is a committed enzyme, because it is going to commit glucose for glycolysis only. You cannot say hexokinase and glucokinase, the very first enzyme, as a committed enzyme, because it doesn't commit that glucose for the glycolysis, but it will commit glucose for the metabolism. So it is not a committed enzyme for glycolysis, right? But PFK1 is a committed, it is catalyzing a committed step for glycolysis. That's what it is. Committed and most regulated step in glycolysis. Most regulated in the sense, it's like if you want to increase the speed of glycolysis, how fast glucose is oxidized into pyruvate, if you want to control that, you got to control the activity of PFK1. If you keep it fast, glucose oxidizes fast. If you keep it slow, it will be oxidation also slows down. It's like your accelerator in the car. You press it hard, car goes faster. Take it back, it slows down. It's like that. So PFK1 is a allosteric enzyme. Allosteric modulators will work on this PFK1 enzyme. So let's see what are the allosteric modulators that will work on PFK1, and which obviously you need to know them. So that is positive modulators. So we have 2 positive modulators and 2 negative modulators. Positive in the sense they are going to work positively with this enzyme and making sure this enzyme is active and converting more and more fructose 6-phosphate into fructose 1,6-bisphosphate. Thereby more and more glucose is oxidized. So negative modulators does the opposite, slows down the enzyme. So if you look at this positive and negative modulation in a allosteric reaction velocity curve, you know that from enzyme lecture, reaction velocity curve for allosteric enzymes, they are sigmoidal, S-shaped, and the positive modulator will shift it to the left side of it, negative modulator shift to the right side of it. Okay, that is what these modulators will do, in case if that is how it is connected. If the questions of enzymes connected with glycolysis, it will be through this enzyme. Positive modulation, negative modulation, left shift, right shift. It is how enzymes can be asked as well. Okay, so AMP is a positive modulator, so it is going to shift the curve to the right side. Fructose 2,6-bisphosphate positive modulator shift to the, sorry, not right side, left side. AMP to the left side, fructose 2,6-bisphosphate to the left side, whereas ATP and citrate to the right side, negative modulator. Okay. Now, why AMP is a positive modulator? What is the meaning? When cell has more AMP, what about ATPs? They will have low ATPs. Always AMP is a hunger signal for a cell. AMP is a product of ATP, right? ATP to ADP, ADP to AMP. If you have more AMP, that means you have less ATP. So more AMP simply means cell lacks energy, so you make more. That's why you break glucose, to make more ATPs. That is why it is a positive modulator, right? Look at ATP here. What if your cell already has sufficient ATP? Why do you need to conduct glycolysis so fast? You slow down. You don't need to go fast. If you left Royal 30 minutes before the class, why you have to rush? You put on the song, come slow. Is it not? Why you need to rush? It's like that. When you have sufficient ATP, you don't need to rush. You have a lot of ATP, slow down, take time. That is what ATP will do to PFK1. It slows down the enzyme, negative modulation. Same concept for citrate. Whenever you have citrate in the cytoplasm, it simply indicates there's a lot of energy for this cell now. You don't need to rush. Slow down. That is what is ATP and citrate means. Now coming to, and look at the location. AMP in peripheral tissues and ATP citrate in peripheral tissues, right? But look at what I have written for fructose 2,6-bisphosphate, which is a positive allosteric modulator, and that is in the liver. Fructose 2,6-bisphosphate is a positive modulator of PFK1 in the liver only. So now, if there is more fructose 2,6-bisphosphate, your PFK1 is most active. Less fructose 2,6-bisphosphate, less active. Is it not? That's what it is, right? Positive modulation means what? If you have more fructose 2,6-BP, your enzyme PFK1 is more active. Less is there, less active. So, who is going to control the concentration of fructose 2,6-BP in the liver? Because liver is a major organ. I told you, right? It's mother. You know, whenever there's a lot of glucose in the blood, who is going to take it? The liver is going to take it, make some glycogen, and make most of it as fat, triglycerides. So, liver has to handle this. So, that means the concentration of fructose 2,6-bisphosphate in the liver matters for glycolysis. So, who is going to control the concentration of fructose 2,6-bisphosphate? Let's talk about two hormones. That is insulin, other is glucagon. Remember, in biochemistry, these two hormones comes every time. Insulin, glucagon, right? What is the role of insulin in our body? Bring down glucose, blood glucose, bring it down to normal, right? What is the role of glucagon in our body? Bring it up. Whenever you are having lack of glucose, bring it up to normal. So they both try to maintain normal glucose levels depending on the situation. If you have hyperglycemia, insulin is going to bring it down to normal levels. If you have hypoglycemia, glucagon will bring it up to normal levels. Both are good hormones. Is it not? Both are good, right? They both try to maintain normal glucose level, right? Now, what do you expect insulin to do to the concentration of fructose 2,6-bisphosphate if it wants to keep your PFK1 high so that glucose is being used, oxidized continuously? Insulin wants to keep glycolysis super active, and it has to make sure PFK1 is working so that glucose is all converted to pyruvate, pyruvate to acetyl-CoA fatty acid and stored. What insulin has to do to the concentration of fructose 2,6-bisphosphate if it wants to keep PFK1 high, means higher, super active? It has to increase the concentration of fructose 2,6-bisphosphate. Is it not? So, insulin, it has to increase the concentration of fructose 2,6-bisphosphate, thereby glycolysis goes faster, thereby more and more glucose from the blood enters into the liver and being used and bring it back to normal glucose. Okay. Now, what glucagon has to do to your fructose 2,6-bisphosphate concentration? It has to decrease fructose 2,6-bisphosphate concentration. Why? Because glucagon wants to decrease PFK1 activity. Why glucagon wants to decrease PFK1 activity? Because glucagon doesn't want glucose to be used from the blood. Because already glucagon means when glucagon is released, only when there is less glucose in the blood. When there is already less glucose in the blood, then you should not conduct glycolysis in the liver. So glucagon has to make sure glycolysis is stopped in the liver. So what glucagon will do? Glucagon will target that PFK1, most active, means most regulated enzyme is PFK1, and the modulator for PFK1 is fructose 2,6-bisphosphate. What glucagon do? Simply decreases fructose 2,6-bisphosphate. Thereby, PFK1 activity decreases, glycolysis slows down. That means entry of glucose into the cell, means into the liver from the blood, also decreases. So you are saving glucose in the blood. Getting this? So at the end of the day, what I want you to know is, what is the effect of insulin on fructose 2,6 bisphosphate concentration in the liver? High or low? High, increase. Thereby keep your PFK1 active, right? Glucagon does the opposite. Glucagon decreases fructose 2,6 bisphosphate, thereby keep your PFK1 kind of inactive, slows down. That is what I try to write here. And the way this insulin and glucagon does this is through another enzyme called PFK2. There is an enzyme called PFK2 which will make this fructose 2,6 bisphosphate. See, this fructose 2,6 bisphosphate, it is made by an enzyme called PFK2. That is phosphofructokinase number 2. Why 2 and why 1? Look at PFK1, what it does. Fructose, PFK1, this enzyme here, PFK1. Where it is adding phosphate? The substrate is fructose 6-phosphate and the product is fructose 1,6 bisphosphate. Where it is adding phosphate? To the first carbon. That's why it is PFK1. It is adding phosphate to the first carbon, right? Now look at PFK2. PFK2, see this PFK, where is it? This is the PFK. I just wanted to simplify the figure. Fructose 6-phosphate is converted into fructose 6-phosphate is converted to fructose 2,6 bisphosphate. That is done by PFK2, which is written in the figure here. This is the green color one, PFK2, which they have written active, PFK2 active, right? So fructose 6-phosphate is converted to fructose 2,6 bisphosphate whenever your PFK2 is active. And who is going to maintain the active state of your PFK2? See this insulin here. Insulin. How insulin is going to make sure your PFK2 is active? So I told you the enzyme that is involved in the formation of fructose 6-phosphate into fructose 2,6 bisphosphate is a PFK2 enzyme. And this enzyme, in fact, it has two enzymatic function. One is the kinase function, other is the phosphatase function. Okay, so let me write that here. Kinase and phosphatase, it has two components to it. This PFK2 enzyme has kinase part, I'll write K here, and phosphatase part, that is P. Kinase, phosphatase. What phosphatases will do? Phosphatase. Kinase. Kinase, what kinases will do? They will add phosphate, right? Phosphatases, what they do? They remove phosphate, opposite, right? So this PFK2 enzyme is in fact is a bifunctional enzyme. It has two functions to it. One is the kinase subunit, other is the phosphatase subunit. Look at the phosphatase subunit, which is put it in like a violet color. They have written it as FBP2, that is fructose bisphosphatase 2. Don't worry about that. Just say phosphatase. Okay. Now this enzyme, this bifunctional enzyme, two enzymes together, it is active, like kinase part of it is active only when there is no phosphate, which is phosphate present over there. Like the surface of this entire enzyme, there is no phosphate added. You don't find any phosphate on the surface. Like I'm going to put these two enzymes together like this, and you don't find any phosphate being added over the surface. That means this enzyme is in dephosphorylated state. Dephosphorylated state means no phosphate present over the surface. Dephosphorylated. Dephosphorylated, no phosphate over the surface of this enzyme. So whenever there is no phosphate over the surface of this enzyme, that is PFK2 enzyme is active. Kinase part of this enzyme is active. As you see, kinase is active. PFK2 kinase active. in dephosphorylated state. And who is going to maintain the dephosphorylated state of this enzyme? It is insulin. How it is going to do it? Always remember in biochemistry, when it comes to regulation, if you are talking about insulin, this is general statement. Whenever an enzyme is activated by insulin, it is always through an enzyme called protein phosphatase. Okay, that is written here, PPP. PPP is protein phosphatase. I'll put this in a simple term as protein phosphatase. Protein phosphatase enzyme is activated by insulin. Always insulin activates protein phosphatase. Phosphatase means removes phosphate. So it is good. This enzyme, once it is activated by insulin, it will go and remove phosphate from the surface of your enzymes. And your enzyme here is PFK2. Phosphate is removed. Once your phosphate is removed, your kinase become active. And the phosphatase subunit, it is inactive. So, because your PFK2 kinase is active, what it does, it's going to take some of the fructose 6-phosphate from glycolysis and make fructose 2,6-bisphosphate. And that fructose 2,6-bisphosphate, it is going to further activate PFK1. So, it is a positive modulator of PFK1. PFK1 enzyme is positively modulated by fructose 2,6-bisphosphate, thereby fructose 6-phosphate is converted into fructose 1,6-bisphosphate and glycolysis continues. That's how your insulin will make sure glycolysis is positively modulated by keeping higher concentration of fructose 2,6-bisphosphate, and that is done through dephosphorylation of PFK2. Okay, that is the concept. And the enzyme that helps in the dephosphorylation process of this PFK2 is PPP. Simply I have written it as protein phosphatase. Protein phosphatase is an enzyme. Okay, this is where you will have some confusion. Okay, let me make it simple. What I have written in the statement. All you need to know is only that statement. Even if you didn't get what I explained, if it is too complicated, you need to know the statement. What is the statement that I have written? Insulin increases fructose 2,6-bisphosphate in the liver by keeping PFK2 kinase active in dephosphorylated state. As long as you know that statement. So your PFK2 kinase is active always in dephosphorylated state. And who is going to maintain the dephosphorylated state of this enzyme? Insulin. This is how insulin works. It always dephosphorylates an enzyme and keep the enzyme active. Most important one, most regulated enzymes in the pathways, if they are activated by insulin, it is always through dephosphorylation. And here PFK2 kinase subunit dephosphorylated, thereby more fructose 2,6-bisphosphate is synthesized, thereby that will have a positive effect on PFK1. So my question here is, does insulin has a direct effect on PFK1? No. It will have an indirect effect. What is that indirect effect? It simply increases fructose 2,6-bisphosphate levels. How it will do it? It's going to keep your PFK2 kinase active. How it's going to do that? By keeping it in dephosphorylated state. That's it. What glucagon will do? It does the opposite. What glucagon will do? Glucagon simply phosphorylates this enzyme. You look at that other side. See the glucagon through protein kinase A. It is simply adding phosphate. Look at this. Phosphate is added. This phosphate. Once it is adding phosphate, you are reversing these enzymes. You are keeping kinase inactive, phosphatase active, and thereby you decrease the concentration of fructose 2,6-bisphosphate. Thereby its positive effect on PFK1 is not there, so glycolysis slows down. So it is just exactly the opposite of what insulin do. So that's what I have written here. Glucagon decreases fructose 2,6-bisphosphate in the liver by keeping fructose bisphosphatase, that is FBPase, what is written there, active, that is phosphorylated state. So remember insulin's function. Opposite is glucagon. If you remember one, you know the other one. Okay. So this is how insulin and glucagon will make sure glycolysis is kept active or inactive by altering the concentration of fructose 2,6 bisphosphate, which is a positive modulator on PFK1. That's how the glycolysis in the liver is controlled. Okay, so that's about regulation of glycolysis. All right, so in this stem, glucose going from 220 to 100. So which enzyme will bring a glucose from 220 to 100? What is the enzyme? Which enzyme responds to higher glucose levels in glycolysis? No, enzyme. Which enzyme responds to higher glucose levels in the in our cells? Yes. You have a couple of enzymes mentioned here. Cyclic AMP dependent protein kinase, nothing to do with here. It's a glycogen metabolism enzyme. And then option C, glycogen synthase, nothing to do with this concept here. Look at B, D, and E. B, glucokinase, D, PFK1, most regulated enzyme, and E, the last one. Now, which enzyme is able to take up as much glucose as possible without being saturated? What is that enzyme? It is the glucokinase. Remember the difference between hexokinase and glucokinase? Hexokinase works at all glucose concentration, but it's going to saturate early. That means it is a low capacity enzyme. Whereas glucokinase, it's a high Km but high Vmax enzyme. It is able to take up as much glucose as possible, trying to bring down glucose back to the normal range. And that's what is done by glucokinase in the liver and beta cells of pancreas. And that's why your answer should be, it is glucokinase. Answer is glucokinase. Only when glucokinase works, then comes PFK1, not before that. That's why those who went for PFK1, your answer should be glucokinase. That is the first enzyme, which is a high Vmax enzyme. Next. So you got to understand the stem first. The compound here that is used, what it does, it decreases the PFK2 kinase activity and increases PFK2 phosphatase activity, right? Remember the bifunctional enzyme, kinase and phosphatase. So if I break this into, say, phosphatase and kinase, PFK2 kinase part and phosphatase part, phosphatase. So kinase, which hormone is going to maintain the dephosphorylated form of kinase? It's insulin, thereby increase fructose 2,6 bisphosphate. And how it is going to maintain the dephosphorylated form of kinase? By activating protein phosphatase enzyme, which removes it. What glucagon will do? Glucagon is going to add phosphate to this enzyme. Say, if you are adding phosphate to the surface, and glucagon always works through protein kinase A, pKa, protein kinase enzyme. Glucagon bind to glucagon receptor, G protein coupled receptor, cyclic AMP increases, adenylate cyclic AMP, protein kinase A. That's how glucagon works. So glucagon always works through protein kinase A, thereby phosphorylate enzymes. And phosphorylation of this PFK2 kinase, it makes the kinase part inactive, phosphatase part active, thereby decrease the concentration of fructose 2,6 bisphosphate. That's why, so if you go with that explanation, so your answer will be phosphorylation by cyclic AMP dependent protein kinase A enzyme. It's a glucagon action. So that is the answer there.